Hello everyone. Today we are going to discuss about duplicate gene and modification of Mendel's dihybrid ratio. Now let us see duplicate genes with cumulative effect. If the dominant alleles present at the two different gene locus are responsible for the phenotypic expression of any trait together, and if these dominant alleles present on the either of the gene locus results in the same phenotypic expression individually, then these are known as the duplicate genes with cumulative effect. One of the example is the coat color in pigs. The parent and generation both having the sandy coat color is crossed. One has the dominant R allele in the homozygous condition and recessive S allele in the homozygous condition, and the other has recessive R allele in the homozygous condition and dominant S allele in the homozygous condition. So each of them produces one type of the gametes. Then these fuse and the F1 generation is all red. Then this F1 generation is self-fertilized and we get the offsprings, 9 having the red coat color, 6 having the sandy coat color and 1 having the white coat color. So the phenotypic ratio here is 9:6:1. So let us see the genotypes of these F2 offspring. Here in the first type where the coat color is red, at least one dominant R and dominant S is present. So dominant R and dominant S is present together, either in the homozygous condition or heterozygous condition. In second type, which has the sandy coat color, either dominant R allele is present or dominant S allele is present in homozygous or heterozygous condition. In third type, both the R allele and S allele is present in the recessive homozygous condition. So from here we can conclude that when both the dominant R allele and the dominant S allele is present, either in the homozygous condition or heterozygous condition, these together produce the red coat color, or these together is responsible for the red coat color of the offspring. But if dominant R allele or dominant S allele is present, either dominant R allele is present or dominant S allele is present in the homozygous or heterozygous condition, then the offspring has sandy coat color. So from here we can conclude that this dominant R or dominant S, when present in the homozygous or heterozygous condition, these individually produces the same type of the phenotype. Hence R and S is known as duplicate genes. And when these are present together, then they produces the red coat color. So these show the cumulative effect when present together. Hence the dominant R and dominant S allele that are present in the two different gene locus are known as the duplicate genes with cumulative effect. When the recessive gene in the homozygous condition present at the two different gene locus produces the same or identical phenotypic expression, then these are known as the duplicate recessive gene. When the dominant allele of two gene locus are present together and they complement each other and results in the different phenotypic expression, then these are known as the complementary genes. Now let us understand this with the example. Two parental generation is cross, both has the white flower color, one has the homozygous recessive A gene and homozygous dominant B gene, other has homozygous dominant A gene and homozygous recessive B gene. F1 generation is heterozygous for both the genes and these are all red. F2 generation has 9 red flower plant and 7 white flower plant. So the phenotypic ratio is 9:7. Now let us understand this 9:7 ratio with the genotype of the F2 generation. In these type of the genotypes, at least one dominant A allele and one dominant B allele is present together. These complement each other and produces red phenotype, which is different from the white phenotype. So how this red color is produced? Dominant gene A is responsible for the chromogen. Dominant gene B encodes for the functional enzyme B, which works on this chromogen and produces the colored product red. So here both dominant gene A and dominant gene B complement each other and produces the red color. In this type of the genotype, at least one gene has the recessive allele in the homozygous condition, and these produces the identical phenotype, which is white flower color. This homozygous recessive allele A and homozygous recessive allele B, these are present on the different gene locus, but these produces the same type of the phenotype. Therefore, homozygous recessive allele of A gene and homozygous recessive allele of B gene produces the same type of the phenotype. So these are known as the duplicate recessive gene. So the dominant allele A and dominant allele B, when they are present together, they complement each other and results in the phenotypic expression red color, which is different from the white color. So these are known as the complementary genes. Now let us see duplicate dominant interaction where the dihybrid ratio is 15:1. When dominant alleles of two gene loci produces same phenotype or same phenotypic expression, and when dominant alleles of two gene loci present together do not show any cumulative effect, then it is known as the duplicate dominant interaction. One example is seed capsule of shepherd's purse, scientific name Capsula bursa pastoris. It has two types of seed capsules, one is triangular shaped and the other is the top shaped. In this, the parental generation, triangular and top shaped is cross. Triangular is homozygous dominant for both the genes and top shaped is homozygous recessive for both the genes. F1 generation we get is heterozygous for both the genes and all are triangular in shape. F2 generation has 15 triangular and 1 top shaped. Ratio is 15:1. Now let us see the genotype of this F2 generation. In this type of the genotype, at least one dominant allele of both the genes are present together. In this type, either dominant allele of gene A is present in homozygous or heterozygous condition, or dominant allele of gene B is present in the homozygous or heterozygous condition. So all these genotypes give triangular seed capsule. Number is 15. So how all these genotypes are giving the same type of the seed capsule, triangular. Dominant gene A encodes for enzyme A, dominant gene B encodes for enzyme B. And these enzyme A and B works on the precursor in the similar manner and produces the same type of the product. So the alleles of both the genes expresses the same phenotype and it is triangular, and these do not show the cumulative effect when present together. So all these genotypes give the same type of the seed capsule, that is triangular shaped. In this genotype, alleles of both the genes are in the homozygous recessive condition and Seed capsule is top shaped. Number is 1. Here, in the absence of the dominant allele A and dominant allele B, and neither enzyme A is present nor enzyme B is present, hence it doesn't function on the precursor and forms the product. So it gives the different type of the phenotype, that is top shaped. In this way we get 15:1 ratio, and this is the phenotypic ratio. So this is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.